Here's a question for you. How many times are you going to check the clock this period, wishing that the seconds will tick along faster so you can get to the next class, and then the next, and then the next? You wish the time will go faster so you can get to the things you want, not caring about the seconds that you miss along the way. But that all changes when someone tells you exactly how many seconds you have left. Every year, the seconds run out for almost 2,000 thyroid cancer patients in the United States alone. All they wish for is hope, that something will put time back on the clock for them. Now, a device like that may not exist yet, but for now, there's Lenvima. Ethan?
So before we go more in depth about Lenvima, we're first going to talk about thyroid cancer. So thyroid cancer is cancer in the large gland that secretes hormones and regulates metabolism. And as you can see here, the thyroid is located at the base of the neck around the esophagus. So our drug Lenvima treats differentiated thyroid cancer, and differentiated means that the cancer cells look and act like normal thyroid cells. So there's two types of differentiated thyroid cancer, papillary and follicular. Papillary thyroid cancer usually occurs in 80% of thyroid cancer cases, and it typically spreads to the lymph nodes of the neck, but while it does metastasize, it grows at a slower rate than follicular thyroid cancer. So follicular thyroid cancer occurs in about 10% of all thyroid cancer cases, and this is more dangerous because it can not only spread to the lymph nodes of the neck, but it can also move to other parts of the body, such as the brain, lungs, and bones. And this is why the prognosis for FTC is not as good as that of PTC.
Okay, so Lenvima was approved of February this year, so it's a pretty recent drug, and it's classified as a targeted therapy drug, which means that it targets specific proteins in cell pathways that lead to cancer functions like angiogenesis, proliferation, and metastasis. It was placed in a priority review program by the FDA, which means that it's approved faster to get on the market quicker, so it can make an impact on cancer patients faster. It's approved for the treatment of DTC in patients that also have radioactive iodine refractory disease, which means that their tumors did not respond to radiation treatment. Now, this is a very dangerous disease, as its 10-year survival rate is only 10%. So Lenvima functions mostly as a drug that extends life. Question?
Yeah, so this targets both of those, the two types of thyroid cancer that you talked about before?
Yes.
Okay.
I'll have questions at the end of this session. So this is the test that the Lenvima's approval is based on, which tested Lenvima versus placebo, which is basically no treatment, in patients with DTC with signs of RRD or refractory disease. So the results were very impressive, as the median progression-free survival rate was 18.3 months, compared to just 3.6 in the placebo group. And after 6 months, 77.5% of the Lenvima group were still survived, compared to just 25.4 in the placebo group. The side effects were pretty consistent with many other cancer drugs. It had similar side effects such as weight loss and nausea that are very common. And the most common serious side effect was hypertension, which is high blood pressure. But that and other serious side effects only resulted in stoppage treatment in a small percentage of cases. Are there any questions about the FDA approval? Yes, John.
Going back to the part where you said like the 80% and 10%. So what is the other type? Is that different type of thyroid cancer?
Oh yeah, there's like multiple other thyroid cancers, but they're
Not as common.
yeah, they're not as common.
So then, what is the rate for? Can you go back to that one? No. Yeah. Okay. So, what is the rate for DTC then? Did you say the other two types? But what is the rate for DTC?
Well, how many people have DTC?
Yeah. Did you say
It's like
It's about these two are the most common types of
those two are
DTC.
referred to as DTC.
Yeah.
Yes. Okay, so no more questions?
I can.
Okay.
So, can you? And one more back. Can you explain? I know that. Can you explain that specific graph?
The specific graph. So this just shows what percentage of the group was still progression-free over this certain amount of time. So, for example, after 12 months, only about 10% of the placebo group were still progression-free, compared to around 60% of the Lenvima group. That answers your question.
progression-free is when the tumor does not
The
grow.
tumor
when
has
it's still
not
there.
progressed. It
Yeah,
hasn't
it's still there.
metastasized or grown. Okay. So now, Jess will talk about that.
Okay, so like we said, the mechanism of Lenvima is the targeted therapy of RTKs, or receptor tyrosine kinases. These receptors activate pathways that result in tumor angiogenesis, proliferation, motility, and invasion. So what Lenvima does is bind to these receptors and inhibit them so that they cannot activate the pathway that will result in tumor angiogenesis, motility, and invasion. So one of those pathways is the RAS-RAF pathway that goes on the class. So as you can see, the RTK is activated, and then that activates this RAS-RAF pathway, which results in angiogenesis, motility, and invasion of the tumor. When Lenvima is introduced, the RTK is inhibited, so the pathway is inhibited as well. So tumor angiogenesis, motility, invasion is inhibited. Any questions? Okay, so Jeremy's going to talk a little bit about some data that supports the.
So this is a Western blot test. Oh,
So,
okay, just wait until the end of this. Okay, end of the session. So this is a Western blot test, which is what scientists use to confirm what the mechanisms that they supposedly put into Lenvima. So this can be a little intimidating, so let me explain a bit. So the E7080 is Lenvima, and this just measures the dosage, so 0.01 microns to 10 microns of Lenvima. And this is P-tyrosine. Now tyrosine is what's transferred from the receptor to the next protein in the pathway to activate it. So if the receptor is actively signaling, then there will be tyrosine residue. Now, as you can see, when there's no Lenvima and the receptor is present, there is tyrosine residue, so it's signaling. However, at doses at and above 1 micron, the receptor is present, there's no signaling, meaning that Lenvima is effectively inhibiting the PDGFR receptor. And these are both inhibited at above 1 micron in both of these different types of cells. So these are also Western blot tests for different types of receptors. In CND, it's the FGF receptor being inhibited, and in E, it is the VEGF receptor being inhibited. And it's very effective at inhibiting this at only 0.01 micron dose. It's effective at inhibiting all tyrosine signaling. So now, question.
Wait, what's E708?
That's just the intro. Any other questions?
I might be missing, but what is exactly what's being shown on the test? It's just the presence of the receptor?
It's just, this is the signaling basically of tyrosine, and this is if the receptor is present or not. So the receptor is present in all the tests, but at and above the 0.01 here, there is no signaling.
So the next thing that scientists tested was the Lenvima's effect on cell migration. So they used a DX3 cell line and a U2OS cell line, which are just other forms of cancers, cell lines, and they were used because they have the same types of receptors as our cancer did. So it should have pretty much the same effect on the receptors themselves. So figure A, they're both showing the same thing. So you can see control, that's just a measure of the tumor spreading and migration. So you can see there's a ton of different lines moving in all sorts of different directions. With just 1 micrometer dosage of Lenvima, it becomes much more concentrated, so there's less cell migration. And with 10 micrometers, it's even more concentrated. So it's not as big of a jump with that bigger dosage, but it is much more concentrated. They also tested the reduced velocity or speed of the cell migration. So as you can see, in 16 hours, the space shown here is completely invaded with cells. But with 1 micrometer dosage, there's space left, so it shows that it's slowed down and it's an even bigger space here. So the invasion reduction was 45% with just the 1 micrometer and an additional 13 with 10. They also tested the inhibition of the cell invasion with Lenvima. So as you can see, the control has 100% invasion, but with 1 micrometer, it was decreased by 57%. So the tumor is still, it's just being much more concentrated with where it is. And with the 10 micrometer dosage, it's decreased by total 87%. So any questions on that?
Is this looking at invasion or migration?
This is invasion. The other two are migration.
Okay, so this is actually like into a tissue.
Yeah. So, Ethan's going to talk now about the
So in order to test this, the scientists used 6 cell lines which represent different tumors, and they performed MTT assays, which are basically tests to determine Lenvima's effect on proliferation. And as you can see in the graph here, there's 6 dose response curves and from which they got the IC50 value, which is basically the measure of how much Lenvima was needed to inhibit cell proliferation effectively. And as you can see here, in the majority of cell lines, Lenvima was only able to successfully inhibit tumor cell proliferation at very high doses, at -6, as you can see. And at these high doses, the side effects would be too severe for the patients to handle. So to summarize, Lenvima is able to inhibit key growth receptors, and this leads to inhibiting tumor cell migration and metastasis and invasion. However, Lenvima was not able to inhibit tumor cell proliferation as well, as this was only seen with high concentrations of Lenvima, which exceed the standard clinical rate. But while Lenvima does not significantly affect tumor cell proliferation, it is favorable for its effective ability at inhibiting cancer cell motility, invasion, and metastasis, as well as its tolerability even after long-term drug use.
Are there any questions on that?
So to conclude, Lenvima is different from other therapies because it possesses a novel type 5 binding mode, which is characterized by its ability to rapidly and potently inhibit kinase activity. And another reason why it's special is because it's a targeted therapy, so most of the side effects of the therapy would go away after treatment ends, unlike chemotherapy or radiation. Any questions?
So, as you say, it's a targeted therapy. So it only targets the 3 receptors, and are those receptors only found on cancer cells, or do they only affect cancer cells?
We actually looked into that. We couldn't really find anything specific to show whether or not it targeted just like the cancer cells or not. So if you want to get back to you, we don't have anything.
Due to the presence of side effects, I assume that it affects some healthy cells, but it's always more effective in getting cancer cells. So what makes Lenvima really important is its effect on cancer patients. Now, according to Cancer.org, right now there are only 6 specifically FDA-approved drugs for thyroid cancer, and only 2 of those are approved for patients with DTC with signs of radioactive iodine refractory disease. Now, those 2 drugs are Lenvima and sorafenib, and when placed in a similar test versus placebo, the difference is clear. Lenvima has almost an 8-month more progression-free survival rate than sorafenib. So for the majority of cancer patients, Lenvima is a huge difference as it extends life by 8 months. And when it comes to cancer, time is everything. Because time is more time to fight cancer, it's more time to survive, it's more time to have experiences. Lenvima is a drug of time. Lenvima is a drug of life. Thank you.
We have about 2 minutes for questions.
Any questions?
Can you just go back to the slide before? Just to clear up, so sorafenib, that was a previous gold standard of treating the cancers.
Yes, that was the previous thyroid cancer. It was made in 2010. So yes.
Do you know how many patients consisted in the Lenvima trial? And the one that you have the graph up there.
Sorry, what was that?
How many patients were in that trial?
How many patients were in the trial in total? It says 261 in the Lenvima group and 131 in the placebo, so 392.
Is that number added risk?
That's the number of risks, so that's the number that still have thyroid cancer. So the reason it went down is either the tumor went away in a small amount of cases, or unfortunately the patients died. Any other questions? Nope. Okay. Thank you.
Get that on camera? Hope so.
You know?